So my goal is to get you kind of energized while you're in your postprandial coma after lunch. And I will admit I changed this talk completely last night because I was thinking about being in the audience, what would I want to know? So, I think the big thing for pediatric neuro-oncology is that we really need a collaborative effort, whether you're an engineer or a chemist or a biologist. And my goal is to really show you how that plays into what I do. And so I'm going to kind of walk you through a sort of day in the life of a neuro-oncologist. But I just want to kind of reiterate 2 points that were made and that Dr. Stiles touched on. And that is the fact that CNS tumors really are the most common solid tumor. So we think of leukemia as the most common cancer, but among those tumors that are non-heme malignancies, brain tumors are the most common. And most importantly, and this was also said this morning by Dr. Yock, is that CNS tumors are the most common cause of cancer death in children. And so that's why it's really important that we kind of work together in this field. So if you're thinking about kind of what do I do in the course of my day, and so initially a patient will come into clinic, and so my day starts off with maybe a patient who happens to have a low-grade glioma, and so this is kind of the representative MRI. The patient may have presented with some signs of diabetes insipidus. They have some visual changes. We were lucky enough to get maybe a biopsy, and it showed that it is in fact a low grade. And I tell the parents that in some ways this is great news. The overall survival is excellent. This patient is going to do incredibly well. But as Dr. Stiles pointed out, it's really an unrelenting, relapsing, chronic disease in the sense that these patients will undergo a significant amount of chemotherapy. Some of them are still often go through radiation, and in that setting you end up with a slew of morbidities at the end of the day and a lot of toxicities for these patients. My next patient that comes in presented with, let's say, hemiparesis, because they happen to have a tumor along their motor strip, which the surgeon was maybe able to debulk but not get out completely. And to these parents, I have to tell them that their child has a type of high-grade glioma and that they may do okay, but not significantly well at the end of the day. And then my last patient of the day is actually a patient who comes in, has sort of progressive difficulty swallowing, maybe some gait abnormalities, significant headaches at a relatively young age. And it earns the patient an MRI in the ER, and what they're found to have is a DIPG. And so diffuse intrinsic pontine glioma is a devastating disease for children. You saw this graph earlier, where in this conversation I'm telling patients that your child is not going to survive beyond 2 years. So where does that leave me in terms of challenges that I face as a clinical neuro-oncologist? And prior to coming to Dana-Farber, I really spent the last 9 years of my 10-year career as a neuro-oncologist looking at clinical trial design and really how can I better design trials that are clinically relevant preclinical trials. So if you think about it, your lab may come up with a great drug. It kills the tumor in the mouse. But the problem is that you cannot achieve a biologically effective dose in a human that doesn't kill the patient. So it may earn you a phenomenal Nature paper that you have this great drug that works in your mouse tumor, but it has no meaning or applicability to the patient in the clinic. That being said, the things that are kind of clinical challenges for me as a clinical trialist and trying to design these sorts of studies are cell lines and accurate mouse models, which was mentioned by Dr. Ligon this morning. So again, for low-grade gliomas, cells don't grow well in culture. And for high-grade gliomas, as an example, which include DIPGs, a lot of times those tumors are incredibly heterogeneous. So if you take a specimen, you may see cells that are typical of a GBM, you may see cells that are suggesting a grade 2 tumor, but that tumor is confined to the pons, which thereby automatically makes it a pontine glioma, and we know that patient's not going to do well. So the limited number of patient samples. So even though low-grade gliomas are, again, the most common tumor among children, a lot of those tumors are not necessarily unresectable or we don't have tumor tissue. By the same token, in DIPG and high-grade gliomas, which are actually rarer, a lot of times those tumors, again, are not resectable, they're in areas that are inaccessible, and so that makes having the availability of tumor tissue to test difficult. Identifying driver mutations, and I think that Dr. Baruakim got to that this morning in terms of really what goes into identifying a driver mutation. If you're lucky enough to identify a driver, can you get a drug for that driver? So one, there may not be a drug available for the target, or two, you have a drug and you can't get it from the pharma company because pediatric brain tumors at the end of the day are not a big business for big pharma. And so we may not be an interest to them unless, of course, they maybe have government funding that they need to consider pediatric indications for their drugs. And so sometimes we're left with actually making the drug ourselves. And then always the question that comes up for us is, does that drug penetrate the blood-brain barrier? And that's going to become important when you go back to modeling as well, because there aren't really any good models at present for blood-brain barrier work. So I'm going to highlight these clinical challenges with two examples, the first being that of the devastating disease of DIPG. And so it's been traditionally diagnosed based on radiologic appearance, and the standard of care is focal radiation, but to call it standard of care really is not correct because it's only palliation, because again, we know that these patients are going to die regardless of the treatment to date we have to give them. So with that in mind, we set out to design one particular trial, which we call DIPG BATS, and I say the next frontier because it really was cutting edge in terms of incorporating biopsy into treatment trials for a tumor that standardly does not get surgery, in which if you're in the clinical community of oncologists, there's still people who consider it a huge no-no to really do surgery in these patients. So the study included patients from ages 3 to 18. We were able to get pre-treatment biopsies. Patients got focal radiation, which again, standard of care combined with bevacizumab, which has anti-angiogenic properties. And then we stratified them by the presence or absence of MGMT methylation and EGFR expression, which we were able to target with commercially available drugs, and I'll get to that in the next few slides. And again, we had 23 participating sites. So how does this all play into this very busy slide? This just kind of gives you an outline of the protocol. But what it highlights is we were actually able to get tissue with biopsy. We were able to increase our limited number of patients, and we had targeted therapies in a sense. So a number of seminal observations helped us to do this. One was work done by a group showing that biopsy in a series of 130 pediatric patients with DIPG actually was feasible, not only feasible, but safe with a transient morbidity of 3.8%. And so we had buy-in from our surgical group in order to do these biopsies. And so this, again, gave us tissue for a limited number of patients. One way to address our challenge. Second was participating centers, giving us the ability to increase our sample size across the board for a relatively rare disease. We were then able to show that biopsies were actually able to supply an adequate amount of RNA and DNA from single cores for testing. And so on average, we had about 5 micrograms and 10 micrograms of DNA, which you notice is actually significantly more than the minimum amount required for genome-wide sequencing. So this showed that we could in fact achieve adequate sampling through a small number of biopsies. Next, if you're trying to design a trial, really the important thing is, can you do these things in real time? So if you're anticipating a treatment and you have to wait 6 weeks for a result in which you're going to stratify that patient, it doesn't necessarily have clinical utility. That patient may progress in that amount of time. And so here you see MGMT methylation was achieved in about a week. And if Dr. Ligon was working hard, we were able to get the EGF expression in about 24 to 48 hours. So let's just review some of those challenges. So one, we actually could address the issue of a limited number of patients with collaborative efforts. We were actually able to get pre-treatment samples based on some work that had been done prior in terms of highlighting the utility and safety of surgical biopsy in DIPG. We were able to identify some driver mutations in a sense that we knew existed from prior work. And then we were actually able to obtain those drugs, but in this case, mainly because they were commercially available. So I'm just going to briefly touch on example 2, and this is really going to highlight sort of all of the issues that come up in terms of clinical challenges in terms of trial design. And I'm not only going to touch on them because Dr. Stiles is going to talk about them in much more detail. But I just want to make a couple points so that as he's giving his talk, you really begin to think about how the interplay of all different disciplines, again, whether it be chemistry or biology or engineering, can really help us move these along and improve cancer care. So the example I'm going to talk about, in fact, check marks the box for development of pathway-relevant models. Actually helps us to elucidate driver mutations for a particular type of disease. In this particular case, we opted to make the drug library, which thereby abrogated any need for an MTA or having to interact with pharma or convince them to give us the drug. We're able to identify drugs suitable for our target of interest, design a method to actually test low-grade glioma in its native state. Again, I said the problem is these cells don't grow well in culture, so how are we going to do this? And identifying a drug that in fact penetrates the blood-brain barrier. So this was mentioned earlier that alterations in BRAF in fact represent the most common drivers in pediatric low-grade gliomas. 10% of those are actually in the form of a BRAF V600E or BRAF mutated form. The bulk of them, however, about 70%, are in the form of the KIA 1549 BRAF fusion duplication. And that's going to become important as we move through this talk from a clinical perspective. So what happened early on, or not early on, but in the last few years, was that we were quite excited when we identified type 1 BRAF inhibitors and found that they were incredibly effective in low-grade gliomas, but these happened to be low-grade gliomas for which patients had a BRAF V600E mutant tumor. So on here you'll see the tumor on the left, for those not used to looking at MRIs, and on the right you see response from the tumor at week 8 of therapy. That excitement, however, was quickly met with the unexpected and unprecedented acceleration of tumor growth in a certain number of patients with low-grade gliomas. So we didn't understand what happened. We had sort of the ideal target. We had a therapy that actually hit that target, and then we saw responses that were unexpected. And what was interesting was that for those patients, these were the patients with the BRAF fusion duplication. And so this highlighted the paradoxical effect on ERK signaling, which will be mentioned in the next talk, and also highlighted the fact that all low-grade gliomas really are not the same. So that then meant sort of looking into what are the mechanistic differences of type 1 and type 2 inhibitors. Since this will be talked about a little bit more, I'm just going to make 2 points. One, that for type 1 inhibitors, they actually recognize their target in the active state. They then bind to the ATP pocket, and they suppress activation of MEK. In contrast, type 2 inhibitors, whether you're looking at the V600E mutants or you're looking at the BRAF KIA fusion, what you see is that your inhibitor recognizes the target in its actual inactive state. It then binds to the ATP pocket in the adjacent allosteric site, and it locks both MEK and BRAF together, thereby presenting downstream activation. And so that became important mechanistically in terms of maybe our type 2 inhibitors will actually work for the majority of patients who don't respond to type 1 inhibitors. So this has since led to the development of a recent trial. This is a phase 1, phase 2 trial that we're hoping to open in the next few months. But it, on the phase 1 part, looks at dose escalation and determination of maximum tolerated dose or recommended phase 2 dosing, so pulling in our pharmaceutical colleagues to look at PK and PD, as well as our pathology department. And then the phase 2 is a multi-strata portion of the study, which includes patients who have fusions with BRAF KIA 1549 or some such similar fusions, patients who have NF1 and low-grade glioma, which we didn't spend a significant amount of time on, but to say that a majority of patients with NF1 do actually have low-grade glioma, and it makes sense from a pathway perspective. And then those patients who have recurrent or progressive tumors that involve anything along the RAS-RAF-MEK-ERT pathway, which was discussed this morning in terms of its importance in low-grade gliomas. And then giving these particular patients our drug of interest, which will be discussed in the next talk, and then continuing based on their treatment response. I think one of the most interesting things about this study is the ability to do a phase 2 with phase 0 target validation. And the idea here is really to look at the patients for whom biopsy is recommended. So those patients may present clinically with symptoms that requires the surgeon to go in and either debulk the tumor or do something to help the patient in that regard. And so for those patients, we can actually give them drug prior to the tumor resection or biopsy, and then take the tumor out and then look at the tumor after the fact. And the goal of doing that is to actually see, are there any on or off target effects? Is our drug present? Because the argument is that our drug gets into the blood-brain barrier. Does this in fact happen when we translate it into patients? And then for these particular patients, the drug continuation is based on whether or not that drug is present in the tumor sample. And just as a final point for that particular study, really the other nice thing about it is that you can begin to look at studies of do studies to look at resistance mechanisms. So for those patients who don't respond across the board, why is it that they don't respond? And the fact that you have tumor tissue that's available pre and post treatment, you can get an idea of why those patients may not have responded. And so just to wrap up, in terms of those ongoing challenges for pediatric astrocytomas and how it relates to what I do, and how really we need an interplay among the people in the room to impact this particular area is, again, in terms of developing cell lines, accurate mouse models, and pathway-relevant models. Acquiring tumor tissue for molecular testing, identifying drivers and patient-derived xenografts. Identifying targeted therapies. Again, determining blood-brain barrier penetration and thinking about models or methods of actually being able to do this. One that's been applied so far is something called MALDI, which will be described in the next talk, which is matrix-assisted laser dosimetry ionization. And it's been an interesting way for us to look at drug penetration before we take our drugs into the clinic. Obtaining or designing drugs of interest. So again, if you can't get pharma to play well with you in the sandbox, how do you get around them? And so do you design the drug yourself? And again, that's at great expense, and so it's not a light undertaking. Methods of drug delivery, and so I didn't really touch on this, but when you think about now convection enhanced delivery, direct delivery into the tumor, are there other methods in which we should be thinking about giving our drugs aside from systemically? So for patients with DIPG, is it better to somehow give that drug directly into the tumor? A lot of this has been sort of the bulk of the work for vaccines and immunotherapy and high grades in adults, and some of which are now being done in pediatrics. I think administering therapies in the setting of standard of care. And so this is something that I've spent the bulk of my work on prior to coming here, and that was if you develop a drug that happens to be a radiation sensitizer and you don't think about in the context of giving it to a patient who's going to get radiation, you may have just set that patient up for side effects of radiation recall. And so again, what you need to do in the lab really needs to preclinically model sort of your clinical goals. And then certainly dealing with unknown toxicities. Again, for the patients with low-grade glioma, they may have excellent outcomes. Their tumors may become quiescent in their 20s, but they have a significant amount of morbidity in their lifetime in terms of toxic effects of chemotherapy and radiation. Whereas the patients, again, with high-grade gliomas and DIPGs have poorer outcomes which need to be addressed. And with that, I'll take any questions or want to talk with the next speaker.